Uh, Thank you so much. So I guess, yeah, this is normally where I would thank the organizers, but I will take the opportunity to thank my amazing core organizer, Shalev, for doing such a wonderful job putting everything together. So yeah, I want to tell you about some work that we've been doing on single cell analysis in cancer. And we've been working in cancer, but the question that we've been very interested in answering is really some of the same questions that we've been interested in answering for the past, God knows how many years. Actually, many ways back to Michael Elowitz's original work in 2002, which is, I'll make it sort of operational for you. If you take one cell and grow it out in a dish. So now you have a bunch of, let's say, cancer cells that are all grown out in the dish. Are all these cells the same? They all look like gray blobs, but you can probably anticipate the answer to this question, which is no. And you can use whatever single cell assay you want, of which we've seen many brilliant examples. They will all show variability from cell to cell. Really, though, the question that I think motivated a lot of this at the very beginning was not so much like, are all these cells the same, but do all of these cells do the same thing? And unfortunately, I would say for the field, if you take one cell, grow it out in a dish, and you have all these cells that look pretty much the same, the answer as to whether all these cells do the same thing is generally yes, they do all the same thing. So, are there really any consequences to these differences? It's been hard to find. And I think part of the reason it's been hard to find them is because well, frankly, I think because we weren't looking at enough cells. And so I put the little asterisk here because that asterisk can be, maybe there's one little cell out down there. Cursor isn't showing up. Anyway, this is a large scale field of view. All these little flecks that you might see in the field of view are actually individual cells, and there are many hundreds of, tens to hundreds of thousands of cells on these dishes. And occasionally you might find a cell that actually does something different from the others. So I'll zoom in on this and show you a little movie of this in action. Here are a bunch of melanoma cells, and I'm going to play this video just to, show you the before picture. So these cells are happily dividing before you add drug. Then we're going to add an anti-melanoma drug to these cells. And what you'll find is that most of these cells end up getting, strung out on drugs. They're like stringy and unhappy and probably aren't going to do very much. But if you are eagle-eyed, you might notice that these two cells here don't look like the others, and these two cells actually started from one cell and even underwent a division. And if I play this video for a little while longer, again, this is with drug applied, you'll see that those two cells keep on dividing and eventually form what is like a drug-resistant colony within these cells, within this field of view, which all begs the question, what was different about this cell to begin with that caused it to behave differently from all the others? And to make a long story pretty short, what we found is that normally people think that when you have a rare cell doing something different, that's an example of a genetic mutation. What we found is that there's non-genetic variability that's strongly associated with this drug-resistant behavior. Each of these dots that you see over here on the left is an individual cell colored by the amount of RNA in them as measured by RNA fish. As you can see to the right, there's one cell that has a very high level, whereas most of them are negative, like that little negative cell on the right. There's one cell that has a ton of mRNA, and it's these rare cells, these like Skittles within the population that express these genes that are the ones that are drug resistant. Now, non-genetic, genetic, what's the difference? One of the key differences is the time scale of reversion. So I actually won't be talking too much about this, but I think it's cool and it's a chance to show a movie. So here's one of these resistance marker genes, NGFR, labeled with GFP. And if you watch it over time, if you watch that cell, you'll see that it goes up and down over time. So it can actually move into the state and then back out of the state. That's part of what makes it a little hard to track down exactly what's going on in these cells because they're constantly flipping back and forth. So what I just showed you is an example of going from distinct molecular states, so different, these cells express these genes, and then linking that to a state in the fate in the future, right, drug resistance. One of the things that we'd really like to do is be able to start with the phenotype, let's say drug resistant cells, and if we could somehow rewind the video, go back in time, say like, oh, okay, here's an individual, the originating cell, what was different about that cell? So the only way you can do this is to use a cellular time machine. And this is the work of Ben Emmerich. I'm just going to go over this pretty quickly because this paper was just published recently. How do you actually make a cellular time machine? So what Ben did, this is a method we call rewind, is you use these lentiviral barcodes that are both in the DNA, but also expressed into the RNA. So we barcode all of these cells by transducing with this virus. We then let the cells divide. So now we've got this like these twins, right? So we've got the orange twins, the green twins, the purple twins, the color of the different barcodes. And then what we do is essentially the twin experiment, like separated at birth. So we take these twins, we separate them at birth, and it's not quite at birth, but you fix half of them for downstream analysis and the carbon copy. So that's like freezing them in time. And then the other set of cells, what we'll do is add drug to them and see which ones form resistant colonies. So in this case, purple is the lucky winner. So the purple barcode grew out and made this purple colony. Then what we do is we say, okay, well, now we know in the future purple wins because we sequence all the colonies and we find there's like the purple barcode. And then what we do, it's a little bit of a crazy experiment. We actually make RNA FISH probes based on the barcodes that we found by sequencing. And using those probes, we can actually light up the purple cell in this carbon copy. So these are the cells that before they even got drug. So this is how you go back in time. We're basically going back to this original population and asking, well, can we find the twins of the ones that went on to become resistant? And then ask, what's different about those cells? And we can do all kinds of different profiling. I really won't go into the details of this too much. I'll probably just show you mostly just some imaging because pictures are cool. Okay, so part of the thing that makes this super challenging is that the frequency with which these cells arise is very, very low. So you really have to look at, many, many thousands of cells. The frequency is 1 in several thousand, depending on the day. But I am pleased to report that after a large amount of optimization, Ben was able to find these essentially needle and haystack cells. You can find these very rare barcoded cells in fields of many hundreds of thousands with high sensitivity and specificity. And they express the markers that we've come to know and love. So, AXL, NGFR, EGFR, and a host of other markers that we were able to discover through sequencing that I won't talk about. But basically, you label this barcoded cell and you can find these other markers co-expressed in that same cell. And you can quantify this pretty nicely. So you can, here's again, AXL, EGFR, NGFR, the y-axis is a number of RNA. Most cells on the dish have very low levels and the pre-resistant twins have very high levels. And, there's a lot more to this that I'm not getting into right now in the sake of time. But one of the things that I wanted to focus on was a really clever question question and technique that Ben used in this rewind paper. So one of the things that he did was he asked this question of whether different initial molecular states could be responsible for different outcomes. And to put this in a little context, when you grow enough of these resistant colonies on the dish, what you'll see is that, well, some of these resistant colonies are big and some are small and some are kind of in between. They're differences. They don't all look the same. So Ben asked this question, well, are the ones that go on to form small colonies the same as the cells that will go on to form the big ones? Is it just, the cell landed in a dish that made it, in the part of the dish that made it small or with certain neighbors that made it big? Or is it all programmed in the initial state of the cell? So what Ben did is he said, okay, well, let's rewind separately the ones that form large colonies from the ones that form small colonies. I won't go through the details of exactly how he could do this, but what he could do then is ask, well, what's different about the cells that went on to form the large ones versus the small ones? And, again, I'm glossing over a lot here, but he could find differences in the initial state of the cell that led to these differences in the ultimate outcome. So what this means is that it's all predetermined. It's all there to begin with. And that determines sort of what you're going to do when you grow up. And this actually really changed how we thought about this problem. Initially, we thought, okay, look, there's some states you become resistant, some states you become dead, right, as a cell. And actually what his results revealed is that there's at least a potential that there's a lot more rich, subtle behavior going on here. You've got some that are become very resistant, and some that seem to become sort of resistant, and who knows what all in between. So this was a question that got Yogesh Goyal, a talented postdoc in the lab, very interested in this. He said, okay, he was just asking the question, how many different resistant types are there? This, very resistant, sort of resistant kind of thing is not particularly systematic or satisfying. So Yogesh did what any modern data scientist would do is, he collected a lot of data, used a neural network to classify all the images, that neural network being his brain, filtering images from his eyeballs, where he just looked in the dish and he just said, hey, look, you can see these different morphological categories of resistant cells. They, some of them seem to kind of grow on top of each other. Some of them look more like fibroblasts. Some are really kind of 3D. Some almost look epithelial. They're these different, and they kept showing up, these like classes of resistant cells. So one of the most important controls that he did early on was ask whether these now seemingly emergent resistant cell types, do they persist through clonal expansion? Essentially, do they breed true? It's actually very much like the old Luria Delbruck experiments. They had to check these exact same things, whether cells bred, whatever. So the way that he would do this, right, is you would take these cells, you add drug, some cells survive, they grow into little colonies, and then what you do is you isolate and expand. So the way you do that, if anybody's ever done this, is you take this little cylinder and you put it on top of the colony and you put some trypsin in it, and it's all very messy and it leaks, and you're like, oh my God, this sucks. But somehow you can do that, and what you'll end up with are these colonies that have grown out. And what he found is that the phenotype bred true. So if they're kind of swirly looking, then you grow them out and they stay swirly looking as long as you can go. The big problem with this, of course, as I've been alluding to, is this is kind of a fussy thing to do, and many of the cells don't really survive this process, so it's really biased, and we don't really have a sense of what is this like ecosystem of resistant colonies. So what Yogesh did is he developed a new technique that allows us to actually in a more unbiased and high throughput way, figure out what are all the different resistant cell types. So he used actually Ben's same barcode system to do this. So what he did was, it's a slightly different design. He barcodes the cells, adds drug, lets them grow. And then the thing is, every resistant colony comes from a single cell that is now grown out and has a particular barcode in it. And then what we do, so now you have cells that have different barcodes all on the dish. They're all resistant cells, but all the cells in a colony have the same barcode. What he would do is mix all these cells together, put them through the 10x machine, and now you get a transcriptome for every cell, but also you do this little side reaction. I won't get into the details of molecular biology, but you do the side reaction, and what you get are, you can link up the lineage barcode to the cell ID from the 10x, and now what you can do is say, okay, this cell's transcriptome, that had the yellow barcode, and this other cell's transcriptome also had the yellow barcode. This one had the pink barcode, and this one also had the pink barcode. So now we have all of that information. But first, let me just show you what you get straight up out of the 10x if you just do one of these UMAP things. What you get is what looks like kind of a ecosystem of resistant cells. I should say this is kind of wild. Like it's kind of interesting to take a step back and think about it. We started with essentially like a single cell on the dish, grow it out, add drugs. All these cells are as homogeneous as can be. You add drug, and all of a sudden you've revealed all this hidden variability. So these differences are real and have sort of systematic and interesting differences here. So for instance, this one big cluster on the right, these are actually SOX10 positive resistant cells. This is something that shouldn't exist, by the way, based on the conventional literature, and for reasons that I'll get into in just a little bit. You can find these ones that look kind of like neural progenitors. There's some over here that look like smooth muscle cells. Other ones that express, all different kinds of markers. So these are really distinct sort of types of cells as far as we can tell, or at least they have a lot of differences to them. Okay, so we also have all this information about the colonies, right? This is just 10x, this is just all the cells mixed together, not worrying about their colony status. So what Yogesh wanted to know is whether the expression clusters that we find match the colonies that, the resistant colonies that we can now identify using the barcodes. So what he could do is paint the barcode. So these are all related cells. You can paint it on the UMAP. And you can see that generally speaking, in fact, almost all the time, what we see is that barcodes map to specific expression clusters. So these expression clusters are really resistant colonies. So you'll see there's, this one over here has a lot, this smooth muscle colony, right? There's this neural progenitor colony. And over here on the right, what you'll see are a bunch of individual cells out there. And that's why these have not been detected before. They're basically single cells sitting on the dish that nobody would ever bother to isolate and be like, oh, look at this resistant like single cell. They'd always focus on the colonies. But there's a lot of these single cells that's sitting on the dish that are actually resistant, and they make up this distinct category of resistant cell. So there's also something that kind of like harkens back to these discussions about Waddington landscapes. And I think, Michael was talking about this idea of potential wells and so forth. I actually think that, we're so used to thinking of potential wells as a way of, cells, cell types kind of push themselves into these wells where they're all next to each other. I actually think that's not true. Necessarily, and it's not what's going on here, at least that much I can say. So for instance, if you look over here, this is this VCAM1 positive cluster. Cluster of gene expression, right? Now we have two colonies that both kind of map to that, right? Like they're kind of both the same resistance subtype. But if you look, they're actually systematically different. They're kind of like next to each other on this UMAP. And you see this for essentially all resistant colonies. It's very rare that they actually really fully enmesh with each other. What this is saying is that all colonies are individuals. Some of those differences between those individuals may be small, some of them may be big, but they're each their own they've all found their own path to resistance. By the way, these differences tend to be ECM proteins, so make of that what you will. Using bulk RNA-seq from these colonies, we can link these morphotypes to the expression clusters. So those also match up pretty well. And, these different types of cells are not just like, oh, they look different. They have very different properties that are relevant for cancer. So for instance, you'll find that they have very different invasiveness. I'm not showing these data, but there's also big differences in proliferation between these different resistant types. And, it's not just some cell culture artifact. If you do these patient-derived xenograft models in mouse, you can find all of these same resistant subtypes appearing in vivo as well. So one of the big conceptual questions that we had, again, is like, are all of these fates predetermined? So Ben's data definitely suggested that, well, at least, some of these fates are very, rigidly typed from the beginning. But now we could ask this question far more generally. So what Yogesh did is he said, okay, well, I'll do this fate map technique. And what I'll do is I'll just take identical twins and do the 10x on the resistant colonies from both, and then ask the question, do twins go to the same place? And amazingly enough, they do. In fact, they fully interdigitate. So it's unlike the colonies that are sort of next to each other. They all really go to the exact same place. They, almost always go to the exact same place. Even these singletons, those like single cells on the dish, they're just because one is a singleton, the twin is highly likely to be a singleton as well, suggesting that, again, these are not just like random single cells sitting on the dish. They're doing something, based on what they were before. And just to, make this point clear, what this is saying is that everything is predetermined. Twins always go to the same place. Even colonies that are different from each other but seem to map to the same resistant type, they're slightly different from each other, but the twins always go to the same place. It's like almost eerie how much everything is all predetermined in this system. So another thing that Yogesh did, which I thought was really interesting, is like, okay, well, that's what this ecosystem looks like at one drug dose. But if you add a different dose of drug, you get many more resistant colonies. You get all different sorts of things going on. What happens when you go to a lower dose? And what happens to the to this ecosystem? And in principle, you could imagine many, many different things going on here. So if to carry this ecosystem analogy a little further, let's say you had frogs, eagles, and squirrels. So one of the things that could happen is that you have the frog turns into a toad at low dose. So you go to a low dose, and what used to be a frog is now some different component. Another possibility is that you alter the distribution. So some things that were squirrels now become eagles. And yet another possibility is that you get, some wholly new thing coming in entirely, like in this case, bats. And what I can tell you is that all of these things happen when you go from high to low dose. We see examples of all of these. So I'll lead you through a few of these. So one example is the component identity shift. So this is like the frog to toad. So you can see over here at the high dose, we have the UMAP has this big bulge over here. At low dose, that bulge seemingly shifts over to the left. And this is actually this melanin A subcluster. Now, of course, without barcodes, it's hard to say, well, okay, well, I have this population, and at high dose, I have this population. At low dose, like, I don't know necessarily that one turned into the other, but because we have the barcodes, we can actually ask, well, does the twin that shows up in this region at high dose, where does its twin go at low dose? And you can actually see that those barcodes almost entirely map one region to the other. So this is an example of something that would become a frog at high dose becomes a toad at low dose. We also see many examples of components appearing and disappearing. So if you look here at high dose and low dose, you'll see here are two patches of things that only show up at low dose. Here's one big patch of things that basically only shows up at the high dose. This is actually, also pretty colocalized with this NGFR expression, this, neural progenitor thing. So one of the things so in principle, these could be either, introduction of a new component or loss of a component, or fate switching, that, something that was going to this part goes to somewhere far different. So what we see actually for this blob over here is that it does do this fate switching. So if we look at the twin, so the twin that at high dose goes to this cluster actually switches more generally to this sort of axial positive resistance subtype. So it actually is switching its subtype. This is an example of fate switching. So this would be the squirrel turning into the eagle. But there are other examples where you can see the appearance of new resistant types. So for instance, if you look over here, we've got this patch that is not there at high dose, it is there at low dose. And if you look at the barcodes over here that correspond to low dose, basically you'll find none of those barcodes show up anywhere at high dose. So this is a completely new population. These are basically cells in the dish that when you give them high dose of drug, they don't survive. But at low dose of drug, they do survive and seem to form some independent or new type of resistant cell that's specifically only there at the low dose. Okay, so hopefully I've been able to convince you that there is actually under this seemingly homogeneous set of cells, if you are looking for the right phenotypes and adopt a sort of phenotype first view of single cell biology, I think what we have found is that there's a large amount of this cryptic variability. And once you know where to look, it's seemingly everywhere. And this variability can emerge or sort of revealed by adding different perturbations. And it depends on the perturbation. So you're going to get different outcomes at high dose, low dose. Different cells are getting selected. Different ones undergo different sorts of reprogramming processes, which I didn't really get a chance to talk about today. We've mostly here, I've been talking about everything in the context of drug resistance, but I think these same principles are going to apply across a wide range of biological processes. And we've already been looking at this in stem cell differentiation and IPS reprogramming. And I think we have some interesting results there too that hopefully I can share with you next. With that, I would like to thank all the people in my lab who I'm so lucky to work with an amazingly talented group of people. Ben Emmert really sort of transformed the lab with his barcodes and working out all these details I didn't have time to go over. Yogesh has really taken it in a super interesting direction. I also really want to thank all our various collaborators. In particular, I want to call out Meenhard Herlin and Ashi Veeraratne. As you might have guessed, I'm not really a melanoma biologist, but Meenhard and Ashi most certainly are. beyond just being, leaders in the melanoma field, they're also just very kind and open-minded people who are willing to listen to kooks like us talk about our single cell, this and that. So, and I want to thank our funders and thank you for your attention. I'll take any questions.
Wonderful talk. Thank you very much, Arjun. We have a question from Siwan Zhang. How much of those differences are driven by genetic variation, mutations, or polymorphism, or rather epigenetic imprinting?
That's a great question. So the variability that we see is, okay, what I can tell you definitively is that the time scales are probably something like 5 to 7 divisions for moving out of a particular, hang on one second, let me get back into the Zoom. The time scales are probably 5 to 7 divisions. Now, that would seemingly preclude things like, SNPs and reversions. I mean, that's like very unlikely to occur on that time scale. Are there genetic things that could occur on that time scale? Sure, like some kind of, episome copy number variation. It's possible. I think it's unlikely for various reasons, including, you don't see like synteny or any of these things that you might expect from sort of large scale chromosomal changes. In a way, it's kind of interesting, right? Like, I think, we're very used to having this need to come up with a molecular mechanism. In a sense, it kind of doesn't matter. The only thing that really matters is the time scale and the reversibility. In terms of what you want to do about it, then yes, I think being able to manipulate it. So we've done CRISPR screens to actually find things that can manipulate these rare cell states. And the very fact that you can manipulate, like we don't find a bunch of like DNA repair mechanisms and pathways that seem to control that. So we do find some, quote unquote epigenetic modulators like a H3K79 methyltransferase. But we also find various signaling proteins and transcription factors. So I would say it's just kind of like run-of-the-mill biology, just hooked up in an interesting way to give rise to this interesting phenotype.
There are many questions, and I'm just going to pick the next one on the list. How do you consolidate this idea of a predetermined difference in the resistant cells with the idea of plasticity between states? Is there some sort of bi, tri, quad stable state that is hard but possible to get out of? And what drives this?
Yeah, so there's a very interesting question also as to whether, I think we're very used to thinking of discrete states, that like you're the gray cell or the green cell, or you're, primed or not primed or whatever. And I think that's actually very misleading. I mean, the results of Yogesh showing that like every colony is its own individual, I think speak to this. Basically, it's a continuum. It's a continuum of states. There's all these different, continuous axes of variation. And somehow that variability can manifest itself in different ways upon perturbation. And it depends on the perturbation, because clearly you're going to end up in different fates depending on which drug or what concentration of drug you give. And that'll either push you one way or the other, So I think it's actually more of a continuum.
Okay, there's a lot of questions, but unfortunately we're running out of time, so I'll just take one more from Matthew Pandelakis. How do you control for the random integration of the lentiviral barcode? Cancer integration has an impact on the drug resistant phenotype.
Sorry, could you say that last bit again? I just
whether the specific random integration of the lentiviral barcode could affect the drug resistant phenotype, the integration points themselves.
Yeah, it's in principle possible. We don't think so, because you can, like if you just look at like the overall resistance proportion of cells that like had the integration versus didn't have the integration, it's pretty much the same. So we don't think that we're getting like some wholesale change. It is possible that individual barcodes are like going into a region that like changes something about the cell that causes it to become resistant, but we haven't seen evidence for any such singular event.
Do we have any more time for more questions, or is that?
I think unfortunately we need to move on, and we'll continue on the discussion forum or the Slack channel.
Okay.